back to Biomindscape. Here the question is, which one of the following enzyme is inhibited by citrate in the Krebs cycle? So here the phosphofructokinase 2, option A, option B is aldolase, option C is pyruvate kinase, and option D is phosphoglycerate mutase. So try to answer this question. Pause and try. Let's move on to the solution. Here, the correct answer for this question is PFK2. Why? It is a key enzyme in a glycolysis and it is inhibited by the citrate, that is an intermediate in the Krebs cycle, and it regulates the metabolic flux between the glycolysis and the Krebs cycle. Here, list of enzymes are given and the functions are given, and we have to tell the correct matches. So cytochrome oxidase, topoisomerase, coenzyme and pCNA. What is pCNA? Proliferating cell nucleus antigen. It is a protein that is associated with which one of the following? Cell cycle, electron transport system, charge link number, and DNA replication. So here pCN, that is proliferating cell nucleus antigen, that is pairing with cell cycle, cohesion is pairing with DNA replication, and topoisomerase, that is pairing with the charge linked number, and cytochrome oxidase, that is ETS, pairing with ETS. Why? This cytochrome oxidase, it is a key enzyme in electron transport chain. The topoisomerase, it is an enzyme, they are the enzyme that changes the linkages number of DNA in the process like replication and transcription, whereas the cohesion, it plays an important role in DNA replication by holding the sister chromatid together. And PCN, proliferating cell nucleus antigen. Proliferating cell nucleus antigen, these are the protein that are associated with the cell cycle in the DNA synthesis and replication. Next question is, the portion of transmembrane protein that cross the lipid bilayer are usually alpha helix, beta sheet, turns and loops. So here the correct answer is, try to answer it, then I'll answer it. It is alpha helix. Yes, correct answer. A is correct answer. It is a transmembrane region of the protein that crosses the lipid bilayer. They are the transmembrane region of the protein. They are commonly formed by the alpha helix. They have an hydrophobic and they can span the lipid bilayer. Whereas beta sheet, it is not correct because it is less common and unusually they are found in the preforming protein. They have hydrophobic in nature. What transmembrane protein? They allow passing through the lipid bilayer and alpha helix structure, the most favorable for spanning the membrane. Welcome back. Here the question we have to solve is the sequence of the template DNA for an mRNA with the sequence. Here a particular sequence is given and we have to tell what is the sequence of the template DNA for an mRNA. So how to find this out? First we have to understand the concept, that is the process of creating an mRNA molecule from a DNA template. What is it called? It is called the transcription. So during the transcription, what happens? The DNA template, it is read as from 3' to 5' direction as given on the screen. To synthesize this transcription, the DNA template strand, it is reading from the 3' to 5' direction, and the complementary mRNA strand, that is from 5' to 3' direction. So base pair rules, these are adenine. Adenine will be pairing with adenine of DNA will be pairing with the uracil of RNA, whereas the cytosine of DNA will be pairing with the guanine of the RNA, and the guanine of the DNA will be pairing with the cytosine of the RNA. So A will be pairing with U, T will be pairing with A, that is T of the DNA, thymine of the DNA, that will be pairing with the adenine of the RNA. So first step is done. Now what is the second step? Here we have the sequence, we have to identify it. So here the sequence is from 5' to 3' direction. That is CA, UU, GG, CC, AA, GG. And here we have to convert it. So we will convert this into C will be getting converted into G, A will be getting converted into T, U will be getting converted into A, G will be getting converted into C, C will be getting converted into G, and A will be getting converted into T here, and G will be getting converted into C. That is from which direction? From 3' to 5' direction. Now we have to reverse this direction. So after reversing, we get CCTT, GG, CC, AA, T, G. That is from 5' to 3' direction. That is standard significant scientific conversion of this. From 3' to 5' we have converted into 5' to 3'. This is a nucleotide sequence and we are writing it from 5' to 3' direction. So we get this sequence, and now let's see which option shows this. So here the option B is correct answer. The following component of lipid rafting in the plasma membrane. So first is cholesterol and B is pignolipid. So here both are found in the they are the component of lipid rafting in the plasma membrane. The lipid rafting are specialized microdomain that are present in the plasma membrane that are enriched with cholesterol and pignolipid. They are important for cellular process like signaling, transduction and protein trafficking. Next, which one of the following enzyme is defective in galactosemia, a fatal genetic disorder in infants? Galactose 1 phosphate uridyl transferase is the answer, GALT, where the galactosemia, it is most commonly caused by the defect in the enzyme GALT. This is an enzyme that is important for metabolism of galactose, and its deficiency lead to accumulation of galactose 1 phosphate, which is toxic to the cell. Here various type of vitamins are given and their disorders which we have to match. Like vitamin A, it shows which one of the disorder here? Night blindness. Vitamin D, fat soluble vitamin. Vitamin C, scurvy, and vitamin K, that is blood clotting. Next, we have to identify a correct sequence of the given intermediate form during the glycolysis. Fructose 1,6-bisphosphate, and we have to arrange them. So first is fructose 1,6-bisphosphate, then fructose 6-phosphate, lactate, and phosphoenol pyruvate. We have to tell which is the correct sequence. So first is A, that is fructose 1,6-bisphosphate, then fructose 6-phosphate, then phosphoenol pyruvate, and then finally the lactate. So A, B, C, D and C, this is the correct order. For glycolysis is a series of reaction, and when the glucose is breaking down into pyruvate or lactate, the ATP and NADPH it is generated. We have to identify the correct sequence for a given enzyme that are involved in the TCA cycle. Aconitis, fumarate, malate alpha ketoglutarate dehydrogenase. We have to tell them. So here first is the aconitis, then D is ketoglutarate dehydrogenase, and after that fumarate, and then finally the malate dehydrogenase is the correct answer. These are the enzyme of TCA cycle, and it acts as a sequence that start with the aconitis, then ketoglutarate dehydrogenase, then fumarate, and then malate dehydrogenase. Here the TCA cycle, it is also known as the Krebs cycle, and central metabolic pathway the oxidized acetyl coenzyme A to the carbon monoxide, that is CO, and then it generates the NADH and FADH. Here the question we have to tell, what are chemokines? Option A, cell surface receptor. Option B, specialized chemotactic cytokine. And option C, any foreign antigen. So chemokine, they are specialized chemotactic cytokine. Why not cell surface receptor? These are the structure. Cell surface receptor, they are structure that are outside the immune cell, that detaches the chemokine rather than chemokine themselves. And any foreign antigen are the substance or particle that are present in bacteria or virus that trigger an immune response. So chemokine are produced by the own body at a traffic directional signal during the response. Chemokines means chemotactic cytokinin. They are small signaling protein that are secreted by the cell, and their primary function includes the chemotaxis that act as a chemical signal to guide the migration. These primary functions is induce chemokines which act as a chemical signal to guide the migration and position of white blood cell leukocytes to specific area in a body during inflammation and infection or injury. Thanks for watching. Do like, share and subscribe with your friends.